So let's begin with review one for lecture exam one. And I have produced a review that reflects my notes primarily. For the reviews for exams 2 and 3, they will be the same thing. So if you need to review, the hierarchy is first use this. And what you should do with the review is take everything out of the notes that you think is relevant and add it to the review. This is what I've done in this review for you, and that's what's depicted in the red font. Now, this won't occur for chapter 5 in this review. You'll have to embellish that one on your own. And also for the future reviews, I won't be doing this either. But I wanted you to see what would be a good review for you to produce so that you're ready for the exam. So what's in black is what will be basically presented to you for the following reviews, and you'll see this in chapter 5. And then what's in red is what I've pulled out of the notes that is important for you to know. My reviews are my exams for the most part. I would say certainly well over 95% of the exam will come directly from the review. I'm not one of these instructors who gives you a review for no reason at all. So I want you to understand that. This review should be your study guide, your production about how to get ready for the exam. That's what I want to teach you. I think that's my job right now, is how to prepare for exams. So let's begin with chapter 1, the levels of organization. You see there, that would be the basic statement that I would have in the review. You should understand there, oh, so I need to add those in here. And so I've listed those for you, the 3 main characteristics of life: reception, metabolism, reproduction. We next define homeostasis, and then the 2 mechanisms of homeostasis here: negative feedback, where you keep changes small within a narrow margin, versus positive feedback like childbirth, and the levels of oxytocin. If you recall our little example there, where you allow the change to go maximal before you get it to go back to near normal. Just as you covered in the first lab quiz last week, or a couple weeks ago, or whenever it was, you will have to know the positional terminology. And so I've defined all of that there, as well as the planes of position. Mid-sagittal plane is important. I explained that. The thoracic cavity, it's divided into the left and right halves by a central mediastinum. I list the organs present in the mediastinum, and then also the abdominal pelvic cavity is found, what organs are found in there. You should know the abdominal pelvic cavity is divided from the thoracic cavity by the diaphragm. The pericardial cavity and pleural cavities occupy the thoracic cavity. You should know the abdominal pelvic cavity is larger than the peritoneal cavity. The peritoneal cavity is a sac within the cavity of the abdominal pelvic region. The peritoneum is a serous membrane. The serous membrane can be divided, it's a 2-layer membrane, can be presented as an inner layer that's called the visceral layer, and the outer layer, that's the parietal layer. So for example, for the peritoneum, you would have the visceral peritoneum that lies right next to the organs, the digestive organs of the abdominal pelvic cavity, and the parietal peritoneum lies next to the body wall. You should know what retroperitoneal means, that it's inside the abdominal pelvic cavity but outside of the peritoneum, and you should know what organs are considered retroperitoneal. Now this first exam will be approximately 50 to 60% matching and multiple choice. So when you see statements like I've presented in this first chapter, most of that material will be multiple choice material. So you study that not to regurgitate it, but to understand it and be able to answer questions about it. In chapters 2 and 3, there are a lot of terms, a lot of definitions, so that lends itself well to matching. So you'll see definitions, and so you should know the definitions so that you can be able to match them to their correct term. And so all the definitions, matter, element, atoms, atomic number, all these definitions you should know in terms of matching primarily. Now, once we get to the big four, we're talking carbon, hydrogen, oxygen, and nitrogen. You should know that those make up 96% of what is you. Your first possible short answer here is to know the atom, to know its basic construct, and I give you an example. It has 5 protons and 3 neutrons right here in the nuclear core. This dotted or dashed line represents the extent of the nucleus that contains the 5 protons and 3 neutrons. This is not a shell, it's just the edge of the nucleus. Your first shell, you should know, can only contain a maximum of 2 electrons. The second shell can contain a maximum of 8 electrons. Since we have 5 total, 5 protons, you know that the number of protons must equal the number of electrons in an atom. So we will have 5 electrons, 2 are in the first shell. That means 3 are left for the second shell. If we consider the valence number in this particular example would be 5 because there are 5 holes, 5 electron vacancies, and our valence electron number would be 3. Our atomic number would be 5 because it's the number of protons, and our atomic mass would be 8 because we add the number of neutrons onto the number of protons. Now, if we were going to consider an isotope of this particular atom, we might look at one that has an extra neutron, which would make its atomic number still 5, but its atomic mass now would be 9 because it has 5 protons and 4 neutrons. But electron number and proton number would not change. Now, these vacancies that exist in the electrons account for the idea that this atom is ideal for bonding with other atoms because it's the electron structures that make up a chemical bond. You should also understand from this picture that the electrons in the first shell have a lower energy, or they are more stable than the electrons in the second shell. The electrons in the second shell are more unstable and thus are more energetic than those in the first shell. You should know about ionic bonds. They're defined above. Ionic bonds then are produced when there is a transfer or the ripping off of an electron from one atom to another. And because of the charge differential now, because one atom will be a positive charge and the other one will be a negative charge, now they are called ions, then we will form an ionic bond because of the opposites attract rule. The second type of bond we talked about was a covalent bond where we share electrons. And if we share those electrons between 2 atoms exactly equally, then that covalent bond is called nonpolar covalent bonds. We saw this between 2 hydrogens, 2 oxygens, 2 nitrogens in a methane group, a carbon with 4 hydrogens. They were sharing exactly equally. But when there's a participant in this bond, in this covalent bond, that does not share equally, that actually hogs an electron or attracts an electron for a longer period of time in its orbit, then the other participant has that electron in his orbit, then we're going to have an unequal sharing of electrons in the development of a polar covalent bond. Water was our example here. We have oxygen having a stronger attraction for the electrons of the hydrogens than the hydrogen atoms or nuclei do for their own electrons. So the electrons of the hydrogen spend longer in the orbit of the oxygen than they do in the own orbit of the hydrogen atom. So because of that, the electrons spend a slightly greater amount of time towards the oxygens than they do around the hydrogens. So we develop a polarity here with the hydrogen side of the water molecule being slightly positive and the oxygen side of the water molecule being slightly negative. Also, this is because oxygen is considered to be electronegative. That means it has a stronger attraction for electrons. Now, when we have a polar covalent bond, we will develop a hydrogen bond. And the hydrogen bond can exist only when polar covalent bonds exist and you have the presence of electronegative atoms. And of course, we used water as our primary example of the existence of hydrogen bonds. But hydrogen bonds will also exist in DNA quite extensively and in proteins as well. This provides an enormous amount of stability to these molecules. You should be able to recognize the way we form large molecules, which is called dehydration synthesis or condensation. We form large molecules, energy-rich molecules, from smaller energy-poor molecules because the energy we're putting in here is all about forming the bonds. We are going to pour energy into the molecule in the form of a bond. And then hydrolysis reactions are when we take these large energy-rich molecules and break those bonds, liberate the energy in the form of hydrolysis reactions. So two examples are given here. You should be able to recognize these. You don't have to draw them, but you should be able to recognize them if I presented them to you. So if you produce water, if it's on the product side of the reaction, then it's dehydration synthesis or condensation. But if you need water for the reaction to occur, it's on the reactant side of the arrow, then you would call that hydrolysis reaction. Our second possible short answer here is to know the four properties of water and what property causes them. The short answers will all be text. That applies above as well to the atom. You will need to be able to describe it in a text format. You can't draw on computers. The tests are computer-based through Canvas, and I will describe to you in another video how you will be expected to take the exam, or I will post an announcement. So the possible short answer here that you will be thinking about how to answer in a text form as well as above, all short answers are that way, is to first describe what are the four properties of water. Well, they expand when freezing. They can form temperature buffers. They're a great temperature buffer, water is. It's cohesive or sticky, and it's also a great solvent. It dissolves many, many things in nature. The property that causes the formation of these particular properties of water is that it expands when freezing due to the enormous presence of hydrogen bonds. It's a great temperature buffer because hydrogen bonds are constantly reforming and forming, and it takes a much higher temperature for these bonds to continuously break. We're talking over 150, 170 degrees Fahrenheit. It's cohesive because water has an enormous number of hydrogen bonds that forms a film on its surface, but it is a solvent because water is polar, and water can dissolve other things that are polar. Two definitions we really have to emphasize here. Hydrophilic is a compound that loves water, dissolves in it freely. Hydrophobic is a water-hating molecule like an oil or a lipid that will not dissolve in it because it simply doesn't have charges. They are made up primarily of nonpolar bonds. We then got into the pH scale, and we defined the zero limit of the pH scale that we're most interested in as that associated with hydrogen or hydrochloric acid. So if you pour hydrochloric acid in water, you can, if you dissolve enough of it, you will form a pH of 0, whereas sodium hydroxide defines the 14 limit of the pH scale. We also defined acids and bases. Acids are those compounds like hydrochloric acid that donate hydrogen ions to solution, whereas bases are those compounds that donate hydroxide or OH ions to solution or accept hydrogen ions. You also should know about salts, not listed here, but just in the back of your mind, keep that in play, that salts are compounds other than those that donate hydrogen ions. In other words, they don't affect hydrogen ion concentration in solution. like potassium chloride or potassium hydroxide. We then started discussing carbon. Carbon is a unique atom that can form 4 bonds because its valence number is 4. Nitrogen can form 3 bonds, oxygen 2 bonds with other molecules or atoms, and hydrogen 1. That's what makes these 4 atoms so great in life. But carbon in particular forms the basis of organic compounds. Now we know also that hydrogens need to be present for the most part, and oxygens need to be present to be called organic compound. But the unique thing, it really sits with carbon. Carbons form long, unbranched chains, but they can also form these branching chains, like in glycogen. They can also collapse on themselves and form ring structures. We'll also see functional groups that are common molecular compounds that form with carbon molecules, such as hydroxyl groups, methyl groups, carboxyl groups, amino groups, and phosphate groups. When we start looking at this whole big picture we've been presenting in chapter 2, we start thinking about the synthesis of large molecules called macromolecules. And remember, they're formed with dehydration synthesis or condensation reactions. We can also break them down in hydrolysis reactions. So if we want to store some energy, we're going to utilize dehydration synthesis compounds and form large macromolecules. But we need to access that energy. We're going to break it down, and we're going to liberate the energy in hydrolysis reactions. So the first of these macromolecules we want to talk about are carbohydrates. Carbohydrates are known for their sweet taste, their energy formation and releasing properties, as well as the fact that they have carbon, hydrogen, and oxygens in a 1:2:1 ratio, such as with glucose, which is C6H12O6 1:2:1 ratio. Simple carbohydrates are one of the first classes we want to talk about, and they can be called monosaccharides. You should know there are two main examples, fructose and glucose. We can combine these monosaccharides into more complex units called disaccharides, and we do that with dehydration synthesis reactions. The disaccharides of interest are maltose, grain sugar, lactose, milk sugar, sucrose, table sugar. The last type of carbohydrate is a polysaccharide. These are your storage forms of carbohydrates. In animals, these storage forms called glycogen, and it primarily occurs in the liver. Its formation occurs in the liver or the skeletal muscle. Also, in plants, the primary storage form of there is starch, but you should also know that it's stored as cellulose, which forms the cell walls in plant cells, and we cannot digest cellulose. We can starch, but not cellulose. The second major type of macromolecule is the lipid. There were 3 subtypes of lipids we talked about. The first are the fats. Fats are made up of fatty acid chains, typically about 16 to 18 carbons long, and they will be attached to a glycerol molecule. A glycerol molecule is 3 carbon chain, and the 3 carbons have hydroxyl or OH groups sticking off of them. Well, as it turns out, all fatty acid chains have carboxyl groups at one end. And go back and review the reaction, but when you take a hydroxyl group from a glycerol molecule and a hydrogen off of the carboxyl group, you liberate water, right? And in the process of dehydration synthesis, then the fatty acid chain attaches to the glycerol molecule and through an oxygen bridge, and you get formation of a fat molecule. The most common of these fat molecules is a triglyceride. That's 3 of these fatty acid chains occupying each of the carbon molecules in the glycerol head. Now, fats can form either saturated fats. This is where your fatty acid chains have all single bonds in between carbons, which means that there are a maximum number of hydrogen molecules present in that particular component. So a saturated fat has maximum number of hydrogen molecules, which means it'll be a solid at room temperature because the fatty acid chains will just lay right next to each other. Unsaturated fats will incorporate a double bond between carbons, meaning they share two electrons. This means then that there will be less than the number of maximum hydrogen atoms attached to these fatty acid chains. And so with the addition of this double bond, this will form a kink in the fatty acid chain, forcing a spacing between that fatty acid chain and the one next to it. This space that occurs then means that the fat will be a liquid. So unsaturated fats are liquids at room temperature and typically much more healthy for you. The second type of lipid we talked about was the phospholipid. It's similarly structured to a fat or a triglyceride, but instead of 3 fatty acid chains, we're only going to have 2 attached to the 3 carbon molecules of the glycerol. On the 3rd carbon molecule on the opposite side from the fatty acid chains, we're going to attach a phosphate group. This phosphate group is charged, hence it can dissolve in water, and this portion of this molecule is hydrophilic. The fatty acid chains are still hydrophobic. So this gives this molecule a dual personality, this phospholipid, where we have part that's hydrophilic, part that's hydrophobic. This is called amphipathic or amphiphilic, and is a hallmark of a feature of the cell membrane that we'll cover in chapter 3. The last type of fat is the sterol. And sterols form cholesterol, which forms all of your sex hormones, your thyroid hormone. So it's a very important class of fats. Their hallmark is that they always have 4 rings. They will have different functional groups sticking off those 4 rings, but you will always see 4 rings in a sterol. Our third type of macromolecule is the protein. You should know all proteins are made from amino acids. There are 20 different amino acids that lead to protein formation in the human body. All amino acids have a central carbon with a hydrogen on one bond, a carboxyl group on the second of the 4 carbon bonds, an amino group on the third of the 4 carbon bonds. And what differentiates the 20 different amino acids is the 4th bond. It has a radical group attached to it. There are 20 different radical groups. The simplest one can be a hydrogen, which is a glycine, or it can be a double ring structure like you see with tryptophan or phenylalanine. There are 4 levels of structure you should know about for proteins, and you should know the first level folding structure is just the straight amino acid sequence. So it's just like reading a sentence. No folding has occurred, nothing's happened. It's just a straight sequence of amino acids. The second level of folding involves the fact that we have nitrogens present, we have oxygens present, they're electronegative atoms, they can form hydrogen bonds. And so hydrogen bond formation accounts for the secondary level of folding, which leads to what are called sheets or helices. Those are the common formations in the second level of folding. A protein is functional at this level of folding. The third level of folding is and this actually applies to this next short answer that we're talking about here. The third level of folding is the tertiary level. The tertiary level involves the interaction of the radical groups. They can often be charged and transfer electrons, and hence we can have ionic bonds form. These are pretty rare, though. There's only going to be a few of these, 4 or 5 or 6 in a protein, so not very common. And finally, the quaternary structure involves the interaction of multiple proteins. Like hemoglobin requires 4 different proteins to interact in order for it to actually be considered a hemoglobin molecule. The other part of this possible short answer is to know the 4 features of enzymes. I'm begging you to know these. There's only 6 words here that you need to know, or 7 words. 6 words. 6 words. I was right in the first place. They can speed up reactions. They are selective, they are reusable, and they're reversible. That's easy to type out. If you want, you can even replace speed up reactions with catalyze, and so you only have 4 little words to know there. You should know that enzymes lower activation energy. Activation energy is the energy required to make a reaction proceed. Enzymes lower that. Remember our little mountain, right? Instead of a big old mountain, we're going to have a little old molehill to climb. The fourth of the macromolecules, or the nucleotides, you should know the structure of a nucleotide. Nucleotides all have nitrogen-containing bases. We'll talk more about those in chapter 4. They must have at least one phosphate group, and they do have a 5-carbon sugar. So the primary nucleotides we'll be talking about in chapter 4 are DNA and RNA that are composed of nucleotides. Of course, they are nucleic acids. But our main nucleotide we're going to talk about in chapter 3, and we'll be talking about it forever here, NANP, is adenosine triphosphate. It is the energy carrier. Whenever you store energy, you're going to take it away from an ATP molecule. Whenever you release energy, you're going to form ATP molecules. The types of reactions that are going to be forming ATP are called catabolic reactions. We're releasing energy. And when we store energy and reduce the amount of ATP present in the cell, we're going to be conducting anabolic reactions. So we'll go to a part 2 video and complete chapters 3, 4, and 5.